Today's activity is a laboratory activity that involves analyzing blood to solve a problem. The lab is titled, Who's Your Mommy? So we have a situation where Kim and Andrea are both new mothers. Both mothers delivered at the same hospital on the same day. Kim gave birth to a baby girl, and Andrea gave birth to two baby girls, which are twins. Each of the mothers took their babies home. However, a couple days later, Kim raised concerns that she believes a mix-up happened at the hospital. Who's your mommy? Today's AP Biology activity involves a lab where we're going to try to determine if a mess-up happened at the hospital. We have a situation where two mothers, Kim and Andrea, both had babies at the same hospital on the same day. Kim gave birth to a single baby girl, and Andrea had twins, two baby girls. Shortly after taking the babies home, Kim raised concerns that a mix-up had happened at the hospital, that the wrong baby was paired up with her, that Andrea took her child home and she took one of Andrea's children home. Your task is to use blood protein analysis to determine each mother and each baby's blood type, and then use those blood types to try to determine if a mix-up occurred and provide evidence one way or the other, either to support a mix-up happened or did not happen. To do this, you're going to need to understand a few things about blood types and blood type analysis. Blood types come in a variety: type A, type B, type AB, and type O. The reason that we have different blood types are because each different type contains a different blood protein. So, for example, type A blood contains the type A protein, and you can see that right here. That protein is commonly called an antigen, and it has a particular shape. A type B protein contains a different antigen, a different protein on there, and has a different shape. Type AB blood contains both the type A and the type B antigen. And then finally, type O does not contain any antigens. No external proteins are located on type O blood. Well, we can use this to analyze blood and to type it. Blood protein analysis is when we check for the presence or absence of different blood proteins by mixing antibodies that will react with the antigens. The presence of a protein will cause clotting to occur. So, for example, if we use what is called the anti-A serum and mix it in with blood type, so if we take blood and we mix in the anti-A serum, if clotting occurs, and clotting is the formation of little particles of blood, that tells us that the A protein is present. So type A blood is present in this blood sample. If we add anti-B and no clotting occurs, it just looks like the normal blood cell, that means the B protein, the B type blood, is not present. So type A blood would look like clotting with the anti-A, no clotting with anti-B. Type B blood would be the opposite, clotting with the anti-B, no clotting with anti-A. If we have clotting in both, we have AB blood. And if we have clotting in neither one, then we have type O blood. So using this information, it's possible to determine an individual's blood phenotype, and in some cases, their genotype. Now, Kim originally took home baby number 3. Andrea took home babies number 1 and number 2. So use your understanding of blood types to determine if a mix-up occurred. The lab and the lab results are posted in OneNote. Since you are doing this activity remote, you can't do the lab yourself. However, I have posted pictures of the lab results so you can zoom in and take a look and determine if clotting occurred. Good luck, and help figure out if a mix-up occurred.